Ryan Collins. Ooh, thank you. Ryan is a recent graduate of Harvard's Genomics PhD program and was a member of Mike Tolkowski's lab at MGH, but he has now moved on to a postdoc at Dana-Farber Cancer Institute. And today he's going to be talking to us about his recent publication, A Cross-Disorder Dosage Sensitivity Map of the Human Genome. So I'll turn it over to you, Ryan. Thank you.
Great. Thank you for the kind introduction, Erin, and thanks also for the invite. It's really exciting to be speaking to you all today and to see a great attendance on the call. So that's always really exciting. Today I'm excited to talk a little bit about one of the last pieces of work I did in Mike Tolkowski's group, like Erin mentioned. I just defended this summer, and so for the slide, I thought it was most relevant to emphasize that until very recently, I was in Mike Tolkowski's group in Boston, and that's where all of this work was done. So for all intents and purposes, this is a collaboration of the Tolkowski group with many of you, including some that are on this call, I see. So that's exciting as well. So I thought I would start by just outlining the motivation for this study at a very high level. And as I'm sure is shared by many of us on the call today, my major scientific interest, and indeed one of Mike's lab's interests over the years, has been understanding it as well as quantifying dosage sensitivity throughout the genome. And so just, this is rote for people on a dosage sensitivity working group call, I realize, but just to really make sure that, the definitions that I'm going to be referring to are kind of consistent with what you all think of in the more clinical spaces. We typically think of two kinds of dosage sensitivity, and this is all at the level of DNA and the genome. I realize there are other definitions of dosage sensitivity at gene expression or protein expression or what have you, but for all of the results I'll be talking about today, it's specifically referring to haploinsufficiency, as in the loss of one or more copies of DNA, but usually just one copy is sufficient to produce a deleterious effect on organismal fitness. And then the opposite of haploinsufficiency is triplosensitivity, which is effectively where the gain of one or more copies of DNA, but again, usually in some of the severe sort of pediatric disorders that we're most interested in this study, I'll be presenting just a single duplication, an additional copy is sufficient to have a damaging effect on fitness overall. We have been approaching this problem of quantifying dosage sensitivity throughout the genome from a variety of different perspectives. And so I think the last time I spoke to, maybe it was this group or maybe it was an affiliated group in ClinGen, but was on our initial release of structural variant calls from whole genome sequencing data in the genome aggregation database. So this was a few years back now, where we analyzed and produced a catalog of structural variants of all sizes and kinds, not just copy number variants and deletions and duplications, in 14,000 unrelated individuals. And we were really interested in building these large catalogs of genetic variation for a variety of reasons, clinical interpretation being one, but dosage sensitivity being the other. But we realized very, very quickly that the sample sizes we have for whole genome sequencing, even to date, but certainly if you go back several years before genome sequencing really started to ramp up in a big way, is that we only had on average one coding disrupting structural variant per gene in 14,000 unrelated individuals in Nomad. And this is extending beyond just copy number variants. This is, exonic insertions or inversions and what have you. And so that really underscores that from a statistical perspective, we are really underpowered at those sample sizes. And so what I'm going to be talking about today is an alternative approach we've used to maximize the availability of genomic data out there through a lot of great collaborations to try and get better statistical power to understand dosage sensitivity in the genome. And I guess I should also mention that I'm happy to be, interactive in this if anyone has any questions, but we can certainly save questions for the end as well. So if there's a burning question, please feel free to interject, or I'll be happy to chat afterwards as well. So if our early genome sequencing studies were underpowered, one data type that we reasoned would be available in much greater numbers and sample size is microarrays, right? So they've been around for a very, very long time at this point. And while they have many technical limitations, we reasoned that they should be reasonably good at, across different array platforms, array CGH or SNP arrays or what have you, if we really were strict about filtering down to very large, rare events, we could get a more harmonized data set. And so what we ended up doing is aggregating large for intents and purposes, here being greater than 100 kilobases, rare being less than 1% frequency. And this frequency was assessed pretty strictly against effectively all available resources you could think of, Nomad, the 1,000 Genomes Project, the Human Genome Diversity Project, genome sequencing data, as well as every one of the array cohorts that I'll be outlining in a second here. So we really tried to be pretty strict about reducing these frequencies or limiting the frequencies to rare in the global population, including every subpopulation in each data set we had access to. And so in total, we were able to aggregate across 17 different sources, which I'll touch on in a second, nearly 1 million individuals worth of copy number variant data. And so this works out to be roughly, 1 rare duplication and 1 rare deletion and 1 in every maybe 4 genomes, something like that. So about every other sample carries 1 such large rare copy number variant ascertained by microarrays. And so we'll talk more about phenotypes in a second as well, because this is I imagine this is a detail that's going to be of interest to the audience today in particular. But we effectively used the human phenotype ontology to standardize the phenotypes across all of these different cohorts. And in so doing, we're able to derive a set of, split the overall sort of meta cohort into a subset of about half, of all samples that met an indication for 1 or more of 54 different disease phenotypes as defined by the human phenotype ontology, and then about half of all samples that, to the best of our knowledge, which again is going to be very imperfect, we'll touch on this in a second, but to the best of our knowledge did not meet any indications for any of those phenotypes. And so then just wanted to pause here and talk about the sources. So like I mentioned, we partnered and collaborated with a lot of wonderful institutions and groups in this work. So this is certainly not just work that we did. And we took advantage of, I would say, 3 main data streams. So one is biobanks. So in here we have the UK Biobank, we have the Estonian Biobank, and we have the BioView Biobank at Vanderbilt University. We also partnered with diagnostic laboratories. So I see some folks from GeneDx on the call today. They were instrumental in getting a lot of, especially for rare disease and pediatric developmental disorder patients, getting, anonymized clinical microarray data into this data set. We also partnered with the Children's Hospital of Philadelphia and Boston Children's and Indiana University. And then in addition to those 2 sort of newer or primary data streams, we also scoured the existing literature for data sets that we felt were, of interest for a variety of reasons, including large sample size, phenotype of interest, well technically QC'd, things like that. And so the full details of all of these sources are in the supplement of our paper, and I'm happy to talk more later. But just to give you a sense of, these are the types of sources that we kind of all aggregated and tried to harmonize in this study. So to go a little bit more in detail into the phenotypes, I can talk about CNV curation if someone has a question on it at the end of this talk, but just for sake of time, I wanted to focus on the phenotypes more than the technical details of the copy number variant calls themselves. So I mentioned we use the human phenotype ontology. Going into this study, we had aggregated a total of 950,000 individuals. And so effectively, each of these individuals had some phenotype information. This ranged from a single phenotype of interest, like in a published case control study, like from the Psychiatric Genetics Consortium, their work on schizophrenia copy number variant association. They had a subset of individuals that were confirmed to be controls for the purposes of psych association studies. And then they had a subset of individuals that met a clinician diagnosis for schizophrenia with DSM-5. But we don't know what we don't know about those individuals. So this is a very rough process, but it ranged from a single phenotype indication all the way up through from some of the clinical groups and genetic testing labs that we partnered with, in some cases, very detailed clinical notes for, indication for referral for genetic testing. And then, of course, the biobanks like UK Biobank, Estonian Biobank have very deep EHR data available on all those samples. So a wide variety of phenotypic information was available here, and we essentially just used a text mining strategy to map the available information we had from each participant onto the human phenotype ontology. And that allowed us to roughly standardize this. It's not perfect, but it did allow us to subdivide the cohort like I'm showing here. And we, after clustering down closely related terms and throwing out some terms for a variety of technical reasons, we landed on a set of 54 HPO terms where each term effectively represents some kind of group of individuals that we know meet the indication for that phenotype. And then we have a set of 500,000 controls that, to the best of our knowledge, which again is imperfect, do not meet the indication for those criteria, for those phenotypes. And so the first thing we did after this rough splitting is we wanted to make sure that the phenotypes that we expected should have burdens of rare copy number variants were indeed enriched for rare copy number variants. And so the first way we looked at this, what I'm showing on the x-axis here is these are each of the 54 disease phenotypes we looked at in this study. The rankings and the colorings will be clear in just one second here. But the first thing we did is we went out into the literature and used 7 different sources, including the ClinGen dosage sensitivity map. We downloaded all of those existing resources from 7 different sources for known genomic disorder loci. So these are loci where large rare copy number variants are known to be associated with one or more human phenotypes, usually severe pediatric developmental disorders. And we then assessed for each of these 54 different phenotypes in our study, if you essentially compute an odds ratio, so this is a fixed effects meta-analysis formally from a statistical perspective, but if you compute the odds ratio, the enrichment of carriers of these rare known disease-associated copy number variants in each of the 54 phenotypes versus the same set of 500,000, approximate population controls, we can see that, across the board, as we would expect, genomic disorder copy number or genomic disorder associated copy number variants do increase your risk for most phenotypes. This is not new. This has been recorded before, even in biobank studies like the UK Biobank. But the phenotypes for which we observe the strongest enrichments are, if you go all the way over to the left on the x-axis here, these are skeletal disorders. This is intellectual disability, growth disorders. These are, what we think of as more severe pediatric onset developmental disorders and structural abnormalities. And so this kind of fits with what we expected, again, suggesting that even though our phenotype information is imperfect and our phenotype matching strategy was also imperfect, it was successfully subsetting groups of individuals that have enrichments for rare, putatively pathogenic copy number variants. We then wanted to ask, okay, outside of the known copy number variant loci that are known to be associated with disease, what is the residual evidence that there's more to find in the rest of the genome from rare copy number variants that wouldn't, be immediately recognizable if you looked at one of the 7 sources that we curated for this set of established genomic disorders? And so to do this, we took a list of genes known to be mutationally constrained against loss of function. This is defined, using a population metric in the genome aggregation database or GNOMAD, but there's a variety of different ways to define such genes, and they all kind of land on the same result. But what we're showing here is the set of constrained genes, loss of function constrained genes from GNOMAD, same analysis as in the top panel, but this time after excluding all known genomic disorders. And so you can see that for deletions, which would match the predicted consequence of loss of function that matches the gene set we're using here, there's a subset of 17 phenotypes that are highlighted in orange where we see particularly strong copy number variant effects from rare deletions of these constrained genes. And by strong, I mean odds ratio of 2 or greater. And so for all intents and purposes in the analyses that I'll present later, we define these 17 orange phenotypes as sort of strong effect phenotypes with strong copy number variant effects. These are mostly developmental phenotypes. And then the rest of them in brown, which do not have an odds ratio greater than 2 for deletions of constrained genes, we kind of treated these as more, it's not perfect, but more adult onset, sort of more modest, maybe more common or complex etiologies. Allowing us to really focus in on the phenotypes that are of greatest interest to our group's work, which is pediatric neurodevelopmental disorders in particular. So after partitioning those phenotypes using our empirical CNV data and the effect sizes we observed, like I said, we identified 17 phenotypes with strong CNV effects and 37 phenotypes with more sort of modest CNV effects. And then, using the HPO, one nice thing is it maps everything onto an affected organ system or tissue. And so this allowed us to subdivide even further by, so among the orange developmental phenotypes on the left, we were able to subdivide using the structured ontology into whether the phenotype of interest is of neurological origin or non-neurological. So I want to emphasize that these are very, very rough phenotypes, but as we'll see in some future analyses that I'll present in a second, they do kind of match up with sort of what we would expect. And so I think we can feel pretty good about these as predictions, certainly not like, gold standard clinical diagnostic or anything, but pretty good from sort of a data mining perspective and genome-wide perspective. So having curated the phenotype data and CNV data for close to 1 million individuals across 17 different sources and 54 different phenotypes, we next approach this data set with a variety of analytical strategies to really understand where in the genome do these rare copy number variants confer substantial risk for disease and why. I'm not going to be able to walk through all of the analyses today, just in the interest of time. And I want to leave, Aaron suggested it might be useful to leave some time at the end of this talk for some discussion and some Q&A. But I'm just going to focus on a couple analyses that I think this group might find particularly interesting. And starting with sort of one of the core analyses we did was we conducted a series of genome-wide association meta-analyses. This is very analogous to sort of rare variant burden testing or your conventional kind of GWAS in common SNP space. But here, instead of using SNP probes, we're using sliding windows of 200 kilobases, where we step across the genome in 10 kilobase increments, every chromosome, masking out regions of low sequence complexity or high segmental duplication and masked regions and things like that, effectively just stepping across the genome, and for every 200 kilobase window, asking statistically, what is the evidence we observe in our aggregated data set that either rare deletions or rare duplications are significantly associated with one of the 54 phenotypes we looked at. And so we did this for each of the 54 phenotypes. I'm showing here just this is a Miami plot. You can think of this exactly the same as a GWAS sort of Manhattan style plot. On the bottom, these are the association peaks we observe for neurodevelopmental disorders. This specific HPO code shown here. For deletions in red, for duplications in blue, and setting a fairly strict genome-wide significance threshold corresponding to a Bonferroni adjustment for all non-redundant tested windows, we arrive at a series of very, very clear peaks. We did this for all 54 different phenotypes, and when you collapse these peaks across all the different 54 phenotypes, we arrive at a set of 178 regions of the genome that we are calling CNV segments. They're effectively, the definition gets kind of a little confusing, and I don't love the terminology, but we couldn't think of a better one. Large CNV segments here, we're defining as a contiguous region where copy number variation, specifically either rare deletion or rare duplication of that entire segment, is associated with one or more diseases at a variety of different statistical thresholds. And I just want to point out that if you compare our results, well, so first of all, when we do permutation testing, we find that these, segments that we land on using a completely blind genome-wide meta-analysis are about 7-fold enriched for the previously reported genomic disorder regions that we curated and used in the last analysis I just presented. And so this is, highly, highly significant, both in terms of effect size and p-value. And so this, not surprisingly suggests that we're landing on a lot of the same sites that have already been well characterized by the clinical genetics community. So that's a good sign that these methods are working and that it's producing results that we actually believe. And then I also want to point out that we did look into sites that have been reported as genomic disorders in the literature, but do not show up at any of the variety of statistical thresholds we applied in our studies. And these are shown with the sort of light circles with the X's through them down here. And you can see that they fall into two broad categories. One is they have extremely strong effect sizes. So, this is an odds ratio of 2 to the 14. It's like almost completely penetrant as best as we can tell. But in our data set, it's also very, very rare. So this might be like 5 or 6 patients out of all million and no controls out of all million samples we looked at. So, these types of loci out on this tail that don't meet our statistical discovery threshold probably are still bona fide disease-associated loci, and indeed probably highly penetrant, but they're just very, very infrequent in the general population, and so are tough to capture from a genome-wide discovery perspective. And then the other category are, a few of these duplications that have been previously reported in the literature as genomic disorders, we do see some evidence for, and the odds ratios are greater than, flat. There's some enrichment in cases versus controls for these sites, but they appear to be also, relatively frequent in controls. And so we think these are maybe more like risk loci, incompletely penetrant, sort of, closer to not common variants, but closer to sort of more like a risk locus that in and of itself wouldn't be the source of a Mendelian dominant phenotype. So those are really the two main categories for why, if you look at the supplement of our paper and a previously reported genomic disorder is not present, it's probably one of those two reasons. And then I should also mention that even if you look outside of known genomic disorder loci, if you take the human phenotype ontology and you map that into OMIM, another nice feature of the HPO terms we used, they can map directly into OMIM, and you can pull all of the genes in OMIM associated with that specific HPO code. If you do that, and then using permutation-based approaches, we were able to empirically assess that these regions that were landing on our CNV disease-associated regions are enriched for genes that have already been implicated in those same phenotypes through independent analyses in OMIM. And so that's, again, just a nice positive control, I think, for these sites. So I'll talk a little bit about the resources of what's in our paper and how we envision it might be useful for the clinical genetics community, at the end of this talk, and maybe another 5 or 6 slides or something like that. But before that, I did want to present just a couple insights that we drew from this analysis and that I think might be informative for how we think about CNVs from like a genome-wide sort of data science perspective. Again, I'm not a clinician. This may vary widely based on sort of your individual clinical bent and individual patients. This is sort of just in general, what are the patterns that we observe? So we took these 178 CNV segments that were associated with one or more of the diseases we considered in our study, and we annotated them with a wide variety of features, and then conducted sort of matched permutation testing by randomly picking random regions throughout the genome to build empirical null distributions of sort of what is the distribution of features that we would expect by chance if you randomly drew regions of the genome that were not necessarily associated with disease. And so the first thing and the most prominent feature is that disease-associated CNV segments are particularly gene dense. So this is on the order of, there are twice as many genes in each of these segments as you would expect by chance if you match on the size of the CNVs themselves. But not only are they particularly gene dense, even if you match on the number of genes in each segment, you still find, particularly for deletions, you still find that these empirically disease-associated CNVs overlap more constraint genes under stronger mutational constraint than you would expect by chance. So these are genes that, in the general population, as defined by, say, NOMAD, if you look up in NOMAD, these are genes that really do not tolerate standing loss of function variation in the general population, presumably due to some, important phenotypic function that the gene plays either in development or, later in life. And then a couple other interesting tidbits from these types of analyses is that really all of our analyses pointed towards it's the genes within the CNV that seem to be, not surprisingly, right? Like that was probably our most parsimonious hypothesis coming into this, but it's nice to see it borne out in the data that it's the genes within the CNV that really define both its phenotypic relevance as well as its penetrance. And so one way of looking at this is if you take sort of as a proxy for penetrance, if you take the odds ratio of each CNV, so this is a relative enrichment in cases versus controls, and you just look at the top third, so these are the most penetrant third of the CNV segments we identified, and you compare them to the bottom third, so these are the least penetrant or sort of more modest effect sort of risk variance that we identified. And then you look at the most constrained gene in those segments, you can see that the CNVs that are highly penetrant are the ones that have genes that are, almost all of them have genes that are under extremely strong evolutionary constraint against loss of function variation in the general population. Whereas, some of these segments, they have, no gene that really is particularly constrained. And, maybe by cause, maybe by, just chance, they also aren't particularly penetrant in the disease risks we identify in our meta-analyses. And then finally, just one other interesting, analysis that I thought was kind of cool that popped out is as a segment grows larger, you can measure this, as a CNV gets larger in terms of either genomic base pairs, so kilobases or megabases or whatever, or in terms of the number of genes in those segments, because they're highly correlated, right? Like a larger CNV will hit more genes. We find, that this trend is quite a bit stronger in terms of number of genes, but it's true for both, and they're so highly collinear, we can't really deconvolve it. But as a CNV segment impacts more and more genes and more genomic space, it is associated with a broader phenotypic spectrum in our meta-analyses. Now, there are some statistical reasons why, we might, question the outcome here, but I think the trends are pretty clear, and it also stands to reason that, it's not hard to imagine that as you impact more and more genes, each of those genes might have a slightly different function or what have you. And so that might potentially explain why some of these CNVs, and particularly some of these famous genomic disorder CNVs, are associated with such a broad phenotypic spectrum and a variable phenotypic spectrum. So if all of our genome-wide analyses pointed to the presence of individual genes within these CNVs being sort of the most relevant actors in the phenotypic presentation of each of these disease-associated CNVs, we wanted to follow this up with an orthogonal data type that might help us refine these regions and identify the critical sort of, you could think of as driver genes or the individual dosage sensitive genes within these CNVs that are most relevant to disease. And this is not a breakthrough idea. This has been done by Evan Eichler's group and Brad Coe and Greg Cooper, and we've also done it in previous autism work with much smaller data size. But here, since we have at this point a wealth of publicly available de novo mutation data from developmental disorder trio exome sequencing, both from the Deciphering Developmental Disorders Project, as well as our most recent autism exome sequencing consortium work, which was just, I haven't updated this slide, but it was just published in last month's issue of Nature Genetics, if you're curious. We were able to aggregate close to 60,000 de novo mutations in children with a broadly defined neurodevelopmental disorder from about 46,000 trios of exome sequence trios where father and mother are both unaffected in the trio. So using these de novo mutations, we then mapped them onto the genes in the CNV segments we identified in our genome-wide meta-analyses. And when doing this and then matching on, the number of genes, the size of the genes, and the gene-specific mutation rates of every gene within these CNV segments, and then you looked broadly at, all deletions, all disease-associated deletions, or all disease-associated duplications, we found a slight trend that deletions in particular are enriched beyond what you would expect under just a background mutation rate for de novo protein truncating variants, but we did not observe this for duplications. Whereas when you look at missense de novo mutations, we found a specific enrichment in duplications, but not deletions. Again, this might line up with maybe mechanism, and this was really encouraging, but we wondered the extent to which lists of, known neurodevelopmental disorder, risk genes might capture this excess. And so we went and from these two studies, the DDD study and our autism sequencing study, we took the genes at meeting exome-wide significance, so a fairly strict list exome-wide significant associated genes with either broadly defined developmental disorders or autism spectrum disorder, and removed those genes from the genome and from this analysis, pretended they didn't even exist in either data set, and then reran this exact same analysis. We found that existing genes that are currently recognized as highly penetrant causes of neurodevelopmental disorders due to point mutations completely explained the enrichment of de novo PTVs in our disease-associated deletion segments. But when we did the same thing for missense variants and duplications, it really did nothing to this enrichment, which I think is both puzzling and as well as really exciting potentially. And this suggests that there is a subset of genes that drive quite a bit of risk for neurodevelopmental disorders through strong effect de novo missense mutations that are not currently accounted for in our established lists of, say, you take your largest exome-wide association study for DD or, autism or intellectual disability. There are genes that are probably acting through mechanisms other than haploinsufficiency, either, maybe gain of function or triplo sensitivity or what have you. So focused analyses on missense variants in future studies, I think, are going to be really well supported by the intersection of duplication data as we see here. I think I probably have time to go through this. I'll go over this a little quickly, but we can come back to this at the end if anyone has questions on this. But just a follow-up analysis we did was, so on this last slide, I presented sort of a global enrichment, right, that we find in all disease-associated deletions. We find an enrichment of PTVs in sporadic cases of neurodevelopmental disorders. And then in duplications, we find the same thing for de novo missense mutations. That only tells us that there is an aggregate burden in those CNVs. So if a CNV impacts 20 genes, we still don't know from this analysis whether it's one gene with all of the excess de novo mutations, or whether there's maybe 3 or 4 genes each with maybe like 1 or 2 more de novo mutations than you might expect by chance, none of which would be wholly penetrant on their own, let's say. So to pick this apart, for each neurodevelopmental disorder associated copy number variant in our data set, we analyzed, or we essentially apportioned the excess of de novo mutations in the exome sequencing data set to each gene in each CNV and rank ordered them to see if there was any patterns that popped right out. And so when you do this for de novo protein truncating variants from the exome data, map them onto our NDD associated deletions empirically in our CNV meta-analysis. And then I'm just showing here, the colors indicate the gene with the greatest excess all the way down through the gene with the 5th greatest excess, going from sort of brightest to darkest. And then all other genes in each CNV are in dark purple. You can see it's quite striking that for most of these regions, almost all of the excess de novo mutations are concentrated in just a single gene. If you sort of percentile normalize this, we can come up with an estimate that 38% of all all neurodevelopmental disorder associated deletions in our analysis have over 90% of their total excess of de novo point mutations explained in no more than 2 genes. So these are likely going to be sort of highly penetrant driver genes, if you will, some of which may be wholly penetrant just on their own. And so, of course, if you just take, say, the top 12 just as sort of a snippet of sort of the CNVs with the strongest enrichments, all of the genes that pop out here are all well-known, dominant neurodevelopmental disorder driver genes. And so none of these are particularly surprising. You can see a couple cases where, there's maybe 2 genes that pop out as particularly prominent, but in general, there's a single gene within each of these deletions that is recurrently mutated in independent patients and is probably driving the neurodevelopmental phenotypes for this CNV association. Now, we did the same thing for missense de novo mutations in duplications, and the picture is a little bit murkier here, which again, I think is really interesting, but also kind of frustratingly inconclusive in a sense. So there certainly are some loci, some duplication loci, where we see a massive enrichment of de novo missense variants in a single gene, but I think that is probably for duplications more the exception than the rule. So here you can see we're only able to explain greater than 90% of the total de novo mutation excess in the exome sequencing data in just 16%, or about, say one in six or so of the duplication associated segments that we were identified in our analysis. And again, if you look at sort of the best candidates here, these are the duplication segments that have the greatest total excess of missense de novo mutations. There's a few genes that pop out here that you might recognize as known to be associated with neurodevelopmental disorders, but the overwhelming take here is that even in the presence of genes like SMARCA2, the known cause of Nicolaides-Baraitser syndrome, or genes like TECNO1H, there are other genes that have considerable excesses of de novo missense variants also in those duplications. And so here I think kind of gives us pause that interpreting duplications is going to be, continue to be a little bit harder, even if you intersect point mutation data and copy number variant data, although I think that's a really promising avenue for those analyses. So just on the last, couple minutes before we get to some discussion and Q&A here, I wanted to present one final analysis we did in this aggregated CNV data set, where we wanted to move beyond defining sets of genome-wide significant associations between one disease and one copy number variant locus. We reasoned that it's going to be imperfect. All sort of machine learning approaches are imperfect. All models are wrong, some are useful. But we reasoned that we probably had aggregated enough data to take a run at predicting the likelihood of dosage sensitivity for each gene in the genome from our aggregated CNV data. And so we approached this as a machine learning problem, where effectively we started by defining, using existing literature evidence, small sets of genes that we had very good confidence were haploinsufficient or triplet sensitive, or tolerant of deletions or tolerant of duplications. And so I'm just showing the criteria for our haploinsufficient and triplet sensitive training sets here. You can see we looked at mutational constraint in Nomad. We looked at sources like Decipher and ClinGen for expert curated associations between haploinsufficiency or triple sensitivity or gain of function mutations in a gene and dominant disease. We also looked in GTEx for population gene expression variation to find genes that were not tolerant of outlier low expressor samples across tissues. And then we also looked at structural variant data in Nomad. And while we're really underpowered at current sample sizes in Nomad, going back to the very first slide I presented, identifying sets of genes that do not harbor any, loss of function structural variant or copy gain duplication structural variants in Nomad is also useful to identify genes that are probably going to be, defined as constrained against copy number variants when we get to larger sample sizes. So taking the intersection of these criteria, we come up with a couple hundred genes in each of these training sets. And then we annotated every gene in the autosomal genome. So we're talking about 18,500 protein coding genes using their ensemble canonical transcripts. We annotated them, each gene, with a suite of 145 gene level features. These are everything from mutational constraint to expression data to GC content to their relative position on a chromosome, being near a telomere or a centromere, things like protein features from uniprot and swissprot. We tried to throw chromatin context. We tried to throw almost every feature we could think of into this model. And then we asked a variety of machine learning architectures in varying complexity, ranging from just like a regularized logistic regression all the way up through a deep neural net. As this model, can you predict the effect sizes we see in our genome-wide meta-analyses from 1 million samples just from their gene level features alone, not looking at the CNV data to make this inference? And the surprising thing was all 8 of these models actually did really well. We were expecting some of these models would not work as well, but when these are receiver operating characteristic curves and precision recall curves, kind of a way of assessing machine learning performance. And so each of the 8 different models we tried is in a different color here. And effectively what these curves are showing is when trained in a cross-validation framework, so we're training on a subset of data and then making predictions on a held out set of data, so we're never double dipping into the data to make predictions on data that we're also training. Each of these models is able to pretty easily classify both our training sets for haploinsufficiency as well as triplo sensitivity, which came as a little bit of a surprise because, again, interpreting duplications has been such a problem, in a variety of contexts compared to haploinsufficiency, where a lot more is known. But we did find that although each of these models performed reasonably well, the ensemble average of all of them actually was the most performant overall. And so that's the yellow model shown here, and that's the one that we used going forward, from which we derived 2 probabilities that we've released as sort of a main component of the resources I'll talk about in a second. The probability of haploinsufficiency, or p-haplo, which corresponds roughly to our model's prediction that a whole gene deletion of that gene would confer substantial risk. We'll talk about what I mean by substantial in one second. Substantial risk for one of the 17 pediatric developmental disorders for which we found, were particularly loaded at the very beginning of the study for, burden of rare copy number variants genome-wide. And then we also defined probability of triplo sensitivity, or p-triplo, which is the exact same thing, but for whole gene duplications. So with these 2 scores in hand, the first thing we looked at was, are they correlated? And the answer was yes. And so from this, we find that our model's prediction is that a gene in general, there are exceptions, but in general, is either likely to be sensitive to both deletion and duplication in the context of disease, or neither. And again, you can see that there are outliers here, in the top left and bottom right corners, but the overall trend suggests that we might actually be able to use existing constraint metrics that have been well defined for, protein coding single nucleotide variants to help prioritize genes that might be sensitive to duplication, just based on this correlation. And then again, we did a variety of quality control analyses just to make sure that we actually believe these scores. So we took a variety of different gene lists. I'm showing just a handful here, and broke them up into bins and asked where are particularly high scoring genes falling in these gene sets for either our haploinsufficiency metrics or our triplo sensitivity metrics. And pretty much, glossing over some details here, pretty much everything kind of lines up with what you would expect, where the gene sets that we would expect based on biological priors, including, the ClinGen dominant haploinsufficient genes we downloaded from you all, are very enriched for high scoring p-haplo genes, and conversely, sort of more dispensable genes like olfactory receptors, or like genes that score as non-essential in CRISPR screens and human stem cells, score quite low in terms of their average p-haplo score. And so this all kind of lines up with what you would expect from sort of a biological prior. And then again, we looked back, we did a variety of different quality control analyses that I won't present all of them here, but one other one I wanted to point out is that this does match up with, population data. So if you look back genome-wide at, all genes in the structural variants in Nomad, we only used a small subset of these genes, like a couple hundred for our training data. So this should be largely independent of our model training. We see that as our model scores a gene more and more likely of being haploinsufficient or triplo sensitive, those genes on average have fewer and fewer loss of function deletions or whole gene duplications in an independent population data set like Nomad. So again, kind of what you would expect. So this is getting closer to sort of how I might imagine, we envision these resources being useful. So one application we looked at was applying this to the copy number variant data that we had characterized from exome sequencing many trios of sporadic autism spectrum disorder. And so this was from the recent publication I mentioned, Fu et al. In Nature Genetics, where this is a completely independent set of about 14,000 children with autism spectrum disorder and 5,000 of their unaffected siblings. We've called de novo copy number variants in those exomes. And if you stratify these copy number variants based on their p-haplo or p-triplo scores, where you're just taking, does this copy number variant overlap a gene with a very high score for either, you can see that we stratify the risk conferred for autism as measured by their, the children, the affected probands and their unaffected siblings by up to an order of magnitude between the top and bottom thirds here. And then again, we also believe that these scores are showing some potential for helping to interpret single nucleotide variants, where we see strong correlations between genes that score highly based on our models for being dosage sensitive and enrichments of both protein truncating and missense de novo mutations in the same data set that I was mentioning earlier. So this is kind of recapitulating what we had already found in our genome-wide analyses. So then the final result I'll show is we realize that these probabilities may be difficult to interpret, and they are not calibrated equally for deletions and duplications. So we wanted to harmonize these in some way so they may be more interpretable and useful for the clinical genetic community in interpretation in an interpretation standpoint. And so to do this, we started by taking all of the genes that are known to be constrained against loss of function variation from NOMAD. We then computed the average effect size we saw in our, the strong effect phenotypes I mentioned, the 17 strong effect phenotypes from the very beginning of this talk. Rare deletions in patients with those phenotypes compared to controls. What was the average effect size of, a more interpretable set of genes that has been, more broadly accepted in the, clinical genetics community, like a constrained gene, finding that the average effect size was about 2.7 for any one of those genes on average. And then we essentially titrated down in percentile bins for each of our, for P haplo and P triplo, and asked, okay, what's the effect size for the top 1% of P haplogenes? What's the effect size of the top 2%? And found the point at which it was a comparable effect size for our dosage sensitivity scores defined based on CNV data compared to existing metrics defined by SNV data in NOMAD. And so using this type of an approach, we can define about 3,000 genes where whole copy loss confers about the same risk for pediatric developmental disorders as PTV in a known constrained gene. Then when we do this for duplications, the first thing you see is that, kind of unexpectedly, on average, the average effect size of a duplication is weaker than the average effect size of a deletion. But nonetheless, we can still define a subset of about 1,500 genes for which whole copy gain confers the exact same effect size as a PTV single nucleotide variant in a gold standard constrained gene. And so I think these 1,600 genes or so are particularly interesting for follow-up in understanding functionally what they do and what diseases they might be most relevant to. So now just talking briefly about the resources and sort of some commentary on how they might be useful for the clinical genetics community, and then leaving some time for Q&A. One thing I want to address sort of right away is that these scores are not completely breakthroughs. I'm not going to pretend that we've solved a longstanding problem that no one else has solved. Indeed, there are many existing constraint metrics and gene scoring metrics out there. And the one thing that I do want to point out that I think many folks are not as acutely aware of is that almost all of these scores are highly correlated. And so I'm showing just a handful here based on SNV data, based on CNV data, based on groups that we've collaborated with, based on completely independent groups, and they all kind of correlate in some way. However, we find that specifically for duplications, but as well as for deletions, our P triplo score provides better, better added value than any individual score on its own. Although probably like a meta score of all of these, or a more comprehensive assessment using all of these scores, might be the way to go clinically, just to look at all the different scores available and make your best judgment on that. And then another way I think I want to mention, I think this study and the data might be useful, is that if you go to our paper and you look in this sort of key resources table, there's a link to this Zenodo repository, which is where we've hosted all of the genome-wide summary statistics from all of the meta-analyses, as well as the scores for all of the genes, the dosage sensitivity scores I just mentioned, and a few other resources, all hosted there for every phenotype. And so this is what it might look like, and you can go there and download the data. And it's a lot of data that you have to unpack and computationally look at. But I think just to highlight how this might be useful, let's just look at two examples. So these are examples that we highlight in the paper, sort of where our summary statistics and our association statistics line up with what's already known about a gene in a given region. And so if you look at the terminal end of 22q, this is associated, deletions here are associated with Phelan-McDermid syndrome, and the known gene is, spoiler alert, is SHANK3 here. What I'm showing down here is above the intersection of the x-axis is the relative frequency of deletions in neurodevelopmental or neurological disorder cases. And in light red, below the intersection of the x and y axes, is that same frequency in controls. And so you can see that these terminal deletions, there's many of them that are quite large, but they all sort of build towards this very tip of 22q. And then, no surprise, when you overlay the genes, you overlay our summary statistics, you overlay constraint from NOMAD, SHANK3 really pops out as the clear driver here, which matches mouse models, primate models. This is the no doubt driver gene of this region. But importantly, SHANK3 would be able to be prioritized just based on our summary statistics alone. So that's the type of analysis I'm talking about, where if you have a CNV of interest and a phenotype of interest, you could try and take a look at our summary statistics and see what genes have the greatest, evidence for association in that region in your patient. And then similarly, just sort of one more example is 17p1.2, another known genomic disorder region. Same story. You can look for the strongest association in this region, and while it's mediated by NAHR, so you can see almost all of these CNVs have very similar breakpoints, there's a handful of patients that have focal deletions of RAI1, which is the sort of recognized driver of Smith-Magenis syndrome, if I'm remembering right, for this region. But this matches, again, known literature on this CNV. And so again, just another example of how our CNV association statistics alone would have prioritized a gene that already has a lot of knowledge about it. And so sort of by the transitive property, you might be able to take a similar approach to CNVs that haven't been as well characterized elsewhere. And then just one last point on how we envision using these dosage sensitivity scores we generated. So these are the p-haplo and p-triplo scores. They're available not only from our paper, but also now Decipher has incorporated them in their suite of resources. So if you're using the Decipher browser to interpret your CNVs, there should be a way to just toggle on p-haplo or p-triplo scores. And just one way that one might use this is if we look at a sample of 34 NAHR-mediated genomic disorder deletions. This is just a subset of those that we curated from the literature. And then if you index into our p-haplo and p-triplo scores for every one of those genes within those genomic disorder CNVs, you can see that the genes that score quite high are genes that you would immediately recognize as being, dominant or near dominant associations with a variety of phenotypes associated with those regions. And so again, sort of just by the transitive property, this type of an approach of if you have a CNV of interest, going and looking at the scores and the summary statistics we find in our meta-analysis might help you to get additional evidence that might help you arrive at a reasonable conclusion for how you want to interpret an individual patient's CNV data or CNV results. And so I'll wrap up there and leave some time for some Q&A. But like I mentioned, this was a massive collaboration spanning many different groups. These are just some of the folks I was able to acknowledge. If you're not on here and you did contribute to this, I'm really sorry. I didn't get you on the slide, but really thankful to everyone who did contribute to this study. And I think we should have a little bit of time for questions.
All right, thank you so much. We have about 6 minutes left for questions. Would anyone like to take the lead? Come off mute, ask your question. Okay, maybe I'll start with a question I have, and apologies if this is naive. So the p-haplo and the p-triplo scores, it looked like they range from 0 to 1, is that correct? And 1 being the most p-haplo and most p-triplo, if you will. Is there a threshold? So if we were evaluating a gene that we have never evaluated before, maybe we don't have a lot of cases. As you know, our evaluations really focus on cases and affected individuals. Mm-hmm. If we're looking at a gene where we don't have a lot of cases and we're trying to make a judgment call, like what is there a threshold? Is it only p-haplo 1 that we should be considering, or is there a lower bound?
Yeah, I mean, so I think that's a really good question. And that is something that we did struggle with in how to sort of frame these results. I think it depends on, again, I'm not a clinician, so I can't really say, the degree to which, what measure of confidence you want. However, I think if you want a comparable data point in how you would treat, because these scores are predictions, right? So they're inherently going to be imperfect and noisy. But if you want a comparator, whatever confidence you give to when interpreting loss of function of a known constrained gene, we've defined thresholds that for p haplo and p triplo that would be analogous to that. So you can assign the same confidence to, so specifically the cutoffs here that we recommend are greater than or equal to 0.86 for p haplo and greater than or equal to 0.94 for p triplo. And the reason that the p triplo cutoff we recommend is a higher cutoff is simply because duplications on average aren't as penetrant. And so, you really need to have more evidence and more confidence this is all in the context of assuming an autosomal dominant model. So you need to have even more confidence in the gene to be more, sure about these predictions.
Thank you. Other questions? I know you guys have other questions. Don't be shy. Erica, I know your group put together a list of questions. Is there one that you would want to prioritize?
I think one question that we are interested in is if it's possible to actually pull out counts for specific recurrent CNV regions because of the way, Ryan, that you did your sliding window analysis. So the recurrent CNV working group has obviously focused on these seg-dup flanked regions, and there are some regions for which we just don't have a lot of data. Mm-hmm. So like this type of analysis with so many data points would be really valuable for us to look at for especially certain regions that we don't have definitive scoring for. So I was just wondering if if it's possible to look at counts for regions like in 22q, there's like the central and distal, all those regions in there, if that's something that we could do.
Yeah, that's a really interesting thought. I think so one challenge that we faced when federating data from so many different sources is we had to operate in a sense of giving up some resolution. We do have the predicted breakpoints from every single CNV, but we can't release individual patient breakpoint coordinates. So that's kind of why we compromised and gave sort of sliding window where, we could infer, cases have, X cases have one or more, or excuse me, X cases have a deletion spanning this window. And then the breakpoints may be beyond that. So there's no identifiability concerns there. I do think there may be a workaround for this in that the biobanks in particular, we should chat offline about,
sure.
Data. But I would say as a stopgap, so 22q is a really tough example. And actually we wrote a whole separate algorithm I beat my head against the wall trying to get a statistical way to prioritize the critical regions. And it worked okay. I mean, it's what we ended up using in the paper. I think there's certainly room for improvement. But if you take a look at regions not quite as complex as 22q, say 16p distal or something like that, for these NAHR mediated, seg-dup mediated regions, CNVs like you're mentioning, the frequency appears to be pretty stable. Like by and large, if you just look at any 200 kb window in that region, we don't see a lot of flux. We don't see in general a lot of sort of smaller focal segmental CNVs within those regions. And so on a case-by-case basis, I think you should just be able to use the summary statistics and take a look at because we provide counts, I believe, per window, as well as normalized frequencies. And so I think that should be a proxy, but let's definitely chat. Feel free to, send me an email offline. And I think there might be a way we would be able to help with this, but I just wouldn't want to, run afoul of any of the concerns with the data privacy and all.
Okay, great. Yeah, thanks so much, Ryan. This was a fantastic talk. Really appreciate it. And we will definitely follow up with you on behalf of the Recurrent CNVs team.
Sure.
All right. Well, we are at the end of our hour. Thank you again so much, Ryan, for this. We have recorded this call. We will make it available on our YouTube channel. We'll send you guys the link afterwards if you'd like to look at it again or share it with your colleagues. And if you have any questions you think of later, please feel free to send them over. I can forward them to Ryan, or you can email him directly. I'm sure he wouldn't mind. I hope I didn't
Please do.
overstep by saying it.
No, please.
All right. Well, thank you guys. We'll talk later.